So here I want to focus on some of the concepts, the bigger concepts of the immune system, and really start talking more about how the innate response and the acquired response works. So we'll first start with the innate response. Now remember, the innate response is the initial line of defense. It's our alarm system. And we have certain cells that are going to be a major factor in alerting your body, your immune system, that there is a foreign invader, there is a pathogen that has entered. And those alarm cells are our tissue-resident macrophages. Now, our tissue-resident macrophages, they reside in all our tissues. Sometimes they're called different names. They can be called the Kupffer cells, for example, in your liver. But regardless of their names, they have the same function. They are phagocytic. They are relatively long-lived inside of their tissues, so they hang out for a long period of time, and they're able to recognize pathogens. Now, we have talked about the innate response, and the innate response, if you remember, is not specific. And what that means, again, is that your macrophage can recognize that there's a pathogen. It can recognize that it's a bacteria, but it can't tell you what bacteria. It can't tell you that it is staph, strep, or E. coli. Just like if it recognizes it's a virus, it can't tell you if it's measles, if it's polio, or if it's influenza. It just knows in general that there's a pathogen there and kind of what category of pathogen it is. The way the macrophage does this is it uses what are called pathogen recognition receptors. So pathogen recognition receptors are on the macrophage. And these receptors are going to identify pathogens via what we call their pathogen-associated molecular patterns. These are also referred to as PAMPs. Now, some pattern recognition receptors are, for example, our toll-like receptors. Now, these toll-like receptors got their name from the Drosophila fruit fly. They were identified in the fruit fly, and they were called toll receptors. Then they were identified in mammalian cells, and they were given the term toll-like because they resembled the fruit fly receptor. So these are toll-like receptors, and they're going to recognize pathogen patterns such as flagella. We do not have flagella, so a PAMP on a bacteria could potentially be a flagella. It could be lipopolysaccharide. Again, we do not have lipopolysaccharide. Pathogens have this, and so that can be a potential PAMP, or pathogen-associated molecular pattern. Peptidoglycan is another one. It can be double-stranded RNA. We don't have double-stranded RNA, etc. So pathogen-associated molecular patterns, patterns of all bacteria, of all fungus, of all viruses, our pattern recognition receptors in our innate response can detect those. And then they can tell that I am seeing something foreign. I'm seeing something that is not self, so I need to get the immune response going. Now, other parts of an innate response that is important are soluble mediators. And so an example of this is complement. Complement are a series of enzymes, and they're called zymogens, which means they are not active until they're activated. Another example of zymogens are your digestive enzymes. They have to get activated in order to work. Same thing with our complement system. So complement is a series of enzymes that are not active. And they bind to, an example of a system within the complement is a mannan-binding lectin. or our MBL system. And this recognizes mannose on bacteria. So we don't typically have mannose on our cell membranes, and so the presence of mannose on a pathogen will trigger the mannan-binding lectin system, which will trigger the complement zymogens and activate them so that they can kill potential bacteria. Okay, so now how does our adaptive response work? So our adaptive response is very different from our innate response. And remember that our adaptive response is going to be specific. And our adaptive response has to learn. It doesn't know, it just has to be taught. And it has to learn what's in your environment. So if you live in an area where Ebola is endemic, your adaptive response will have to learn about Ebola. If you don't live in an area where Ebola is endemic, your adaptive response doesn't learn about it. If you live in an area where you have polio endemic, your adaptive response must learn about that. Same thing with flu, etc. So your adaptive response tries to get to know what pathogens are in the environment so that it can develop an immune response to those pathogens, because the likelihood, again, of encountering them again is going to be much higher than if the pathogen is not in the environment. So this leads to what is referred to as anticipatory adaptive clones. A clone is just an immune cell that makes copies of itself. So cells like to replicate, cells like to divide. A hepatocyte is a hepatocyte and makes other hepatocytes, and they all look the same, and they all have many similar functions. And it's the same thing with your immune system. If your immune system decides that Ebola is in the area, it wants to make a lot of T cells and B cell clones that can respond to Ebola. And so remember, our adaptive response are specific, so it's a specific cell targeting a specific pathogen. Those cells are our B and T cells only. Our B cells make antibodies. Our T cells are our cell response. And so we have some levels of anticipatory concepts. So we first have the genome. So the genome of B and T cells enable B and T cells to generate millions of antigen receptors. And these are all different. And so for the B cell, this is called the B cell receptor. And for the T cell, it's called the T cell receptor. And so this is carried out by recombination events. These lead to diverse receptors. Now what recombination means is that you have a gene that combines with another gene. Recombination means you have many genes that can mix and match with many other genes. And these are going to generate those receptors that will recognize the different pathogens that are in your environment so that the B and T cells can learn and respond to those pathogens. So we have our germline. Germline is what you inherit. So this is what you get from your mother and from your father. You're going to inherit germline DNA. And germline DNA is going to be important for your adaptive immune response molecules. So it's going to be important for those MHC molecules, major histocompatibility. And this is a complex. For your T cell receptor, for your B cell receptor. And so those are all going to be inherited. So you are going to get from your mother and from your father copies of their genes, and these genes are going to be codominant. And this is important, which we'll talk about diversity in a future talk. Now, there's also somatic changes that can occur, but this is only with respect to B cells. And I don't want to spend too much time on it right now because this is just sort of a sweeping overview again. And when we start talking about B cells more specifically, we go into how, at the somatic level, they can change themselves. So this is only with B cells and antibodies. And the purpose is to get better at binding. So just as a quick little side, let's pretend we have a triangle antigen. So here's our antigen. This might be a flu virus, it might be a staph infection, but that is our antigen, and that's what the B cell receptor will see. And so we have our B cell receptor. Let's say our B cell receptor looks like this. So here's our B cell receptor, the N. So this is our Fab region. If you don't know what Fab means, please watch the previous lecture. And so for somatic recombination, this Fab region will now change. The antigen doesn't change. It still stays as this lovely little red triangle. But what happens is that your B cell receptor now becomes better fit at binding to that antigen. So instead of being this sort of roundish circle, it now has sharper edges similar to the triangle. And so that's occurring at the level of the somatic mutations that can occur only in B cells and only with respect to their antibodies. Now, another very big, broad, sweeping concept that's important to understand with respect to your acquired immune response is clonal expansion and clonal selection. And I use acquired and adaptive interchangeably. So anytime you hear that, only think of B and T cells. Those are the only two cells that can do this. Okay, so we have clonal expansion. Expansion is easy. Expansion, you expand. Clonal selection is going back to that specificity. So we have a whole bunch of B cells. We have a black B cell. And we'll say on its surface it has an antibody, which looks like a lovely little Y. We have a green B cell. It too has on its surface a green B cell receptor. And let's say that this B cell is sort of curved. Go back to this guy and say that he has like a V-shaped. And we also have a purple B cell. And this B cell has, again, a B cell receptor on its surface, and it looks like a square, purple square. And so when you get infected, you have some sort of pathogen, and we're going to say it has these green little circles. Okay, and so the B cell that gets selected will be this little green guy, the green B cell. And so that is selection. Now the second part that comes in is going to be expansion. Oops. Now all that means is that this green guy is going to expand. And this expansion, the green guy will still only have on its surface the B cell receptor that has the little cup shapes. All of these guys, I'm not going to draw all of them. And they're going to release antibodies. Oops. Antibodies are free-floating B cell receptors, basically. And they still have their little cup-shaped, all of them. So they'll look like the B cell receptor from the B cell they were from. And they're going to continue to bind to those green circles. Okay, so this is selection, expansion, and from this expansion, you will eventually get memory. And so memory, of course, is what you ultimately want from your acquired immune response. And this same selection expansion happens with T cells as well. They are going to respond to peptides of this green pathogen, and only the T cells that respond to those peptides will be selected for, expand, and then form memory. Okay, so let's try to tie a little bit together our innate response. So I have here this lovely foot, and this lovely foot is going to get cut. So we have a cut, and then we have some damage and bleeding, and part of that is that a bacteria gets inside. So we have our bacteria. Oops. So that little blobby thing is our bacteria. Okay, so we got cut, got exposed to this bacteria, so who's there to respond and alarm the system is our macrophage. So we have our blobby macrophage. Here's our macrophage. And our macrophage has a toll-like receptor on its surface, and it's going to recognize flagella, for example. And so it's going to be able to bind to this little flagella. We'll say these guys have flagella. And the toll-like receptor will activate the macrophage. And the macrophage will alarm. And so what does this alarm system do? The alarm system is that your immune cells have a way to communicate with each other. So they're going to release these proteins that are going to help the immune system figure out there's an infection in the big toe or the foot. And they're going to tell your immune cells, you guys need to go to the foot. And so, of course, in your foot, you'll have your endothelial cells that are lining the blood vessels. So here's your endothelial cells. And as you get closer to the site of infection, these endothelial cells get kind of leaky, so they kind of spread apart. And so this allows your cells to enter. So in the blood, you'll have your white blood cells. Maybe I'll make this guy blue. So you'll have your white blood cells, and then these guys will enter. So this is going to be, for example, a neutrophil. So here's our neutrophil. Now our neutrophil will come along, and our neutrophil will start to help our macrophage. Neutrophils have bilobed nuclei. Our neutrophil will help our macrophage by eating the bacteria. So it's a phagocytic cell. The macrophage eats the bacteria. It's a phagocytic cell as well. And so you have your neutrophil now recruited to help this macrophage fight off this infection. At the same time, you have another cell here, which is also a tissue-resident cell, and it's our dendritic cell. So dendritic cell here. Now our dendritic cells are kind of chicken. They are going to pick up this bacteria, but they run. They're going to head out, so they take off. And so our dendritic cell picked up this bacteria with its flagella and took off. Our macrophage stayed, our neutrophil stayed. The other thing that we have going on at the site is we have our complement, our mannan-binding proteins that are going to try to kill the bacteria. So we also have that. Now what our dendritic cell is doing, so let's trace our dendritic cell. Our dendritic cell is actually going to a lymph node. So our lymph node, we can think of it as like a little kidney bean. You have a medulla. You have a paracortex and a cortex. So you have your B cell zone. You have your T cell zone. I should have made that bigger. Let me try to make that guy bigger. And so your dendritic cell is really going to be there to help your T cells. So your dendritic cell with the purple bacteria, with its MHC molecule on the surface, it has the purple peptide. So we have our MHC is going to interact with our B cell, or a T cell. So here we have a T cell with the T cell receptor binding to the peptide associated with the MHC molecule. And then this T cell gets selected for, and this T cell will expand. And the T cell will go back to the site of infection, to the toe, and will help out. So now we have our T cell going there. Now our B cell is a little bit different. Our B cell does not need a dendritic cell. So we'll have our B cells, and they'll have their B cell receptor on the surface, and their B cell receptor is going to bind to the bacteria directly. There's a little flagella, and our B cell is going to get activated. And you get clonal expansion, and then you get antibodies released, and the antibodies will go back and start to help. And so all of this is sort of going on in a sequence. And once you get the acquired immune response going, you're going to be able to clear the infection much faster because it's going to bring a lot of tools into the site of infection. Okay, so let's talk about our antigen presenting cells. Now, our antigen presenting cells are critical for our acquired immune response. Our antigen presenting cells, in essence, is a cell that can activate a CD4 T cell. Notice I don't say CD8. And so the question is, why? Why CD4? Why not CD8? Well, we have our major histocompatibility complexes. And they're called MHC. And we have two versions of MHC. We have MHC class 1, which we call MHC1, and we have MHC class 2, MHC2. MHC class 1 is on all nucleated cells. So one cell that has no nucleus is the red blood cell. So an MHC1 is not on a red blood cell. MHC2 is only on APCs. So the only cells that can interact with a CD4 T cell is going to be your macrophage, your dendritic cell, and your B cell. So MHC class 1 activates CD8. MHC class 2 activates your CD4. Okay, so a way to look at this is you have a CD8 cell, you have a CD4 cell, you have a hepatocyte, liver cell. It has MHC class 1, and you have your CD8 TCR, you have some sort of peptide, and you can get activation. For your CD4s, this has to be, for example, a macrophage. You have class 2, you have your TCR, you have a peptide. Class 2 tends to be a little bit longer, so I'll make it a little bit longer looking, and you can get activation. So the big thing to remember here is that there are two types of MHC molecules. There are class 1 and class 2. Class 1 is expressed everywhere, and class 2 is only expressed on 3 types of cells: your macrophage, your dendritic cell, and your B cell. And so that limits the amount of information that your CD4 T cell can get. Remember, your CD4 T cell is your helper cell. And so it sets up the immune response. Your CD8 T cell is your cytotoxic T cell. So the cytotoxic cell has to kill an infected cell. The helper cell just needs to coordinate the immune response. And you're going to control that coordination by limiting how or what cell that CD4 T cell can actually interact with. Okay, so next up, let's talk about the phases of an immune response. This is one of my favorite things to talk about because it really drives home the importance of vaccines, which we will discuss at a future time. But so you have phases of your immune response. So you have time, and you have on this axis magnitude. So in essence, you'll be infected with a pathogen, and that pathogen will infect you. Usually pathogens go into cells or they have to replicate in order to be detected at large numbers, and then you will clear that pathogen. Now this typically is an acute infection. There are, of course, exceptions to this with chronic infections, but we're not discussing that here. This is just the phases of a normal immune response. So you first have up your innate response. So we'll draw this here. So here's your innate. And this is sort of a speed bump. This is that alarm system, your complement, your macrophages, tissue-resident macrophages, recruiting other cells, etc. So that's all going on in your innate response. You will also have an antibody response. So your antibody response will come up. And your antibody response is usually poor initially, and then your antibody response gets better. Your poor antibodies are going to be IgM, and your good antibodies, for example, will be IgG. It could be IgA, it could be IgE, it depends, but it's typically not IgM. The first antibody is always IgM, and it's always poor. Now you can see that your good antibodies come up, and it takes about 2 weeks. So most pathogens are already gone before you even get your good antibodies. Now the T cells are going to come up, and your T cells usually come up, and then they're going to clear the infection, and then they're going to form memory. I'll just put T cells. So really your T cells are what is controlling the infection. And so what do we call these stages of the immune response? We have the initial stage here, which is really called activation. And then we have the next phase here, which is really the expansion. And then you have another phase here that's referred to as contraction. That's when everything is cleared up. And then these two lines here form memory. And so those are really the phases of the immune response. All right, so we've talked about our innate versus acquired immune responses. And there's another concept with respect to your acquired immune response, which is active versus passive. So active means you developed a response. Passive is just that you were given a response. And this could be a baby from the mother to the baby, which is referred to as maternal antibodies. This could be serum antibodies from an exposed person to an infected person. So the exposed person was infected at one point, developed antibodies, and you can take those antibodies from that person who recovered from the infection, transfer it to a person who is suffering from the same infection, and provide passive immunity. An example of this is rabies. And there's also lab-acquired monoclonal antibodies. Same concept as that rabies, except for now they're artificial antibodies made in a lab, but it still provides some sort of passive or given immunity to help a person who's suffering from a disease such as rabies that would kill them before they were able to control the actual infection. Okay, and lastly, just a little bit on those helper cells. So I keep saying that they're helper cells. We know that they can only interact with, so our CD4s only interact with our macrophages, dendritic cells, and B cells. So what is really going on with these helpers? So I want to bring up a few different types. We have different types of helpers. We have a type 1 helper, which we call Th1. We have a type 2 helper that we call Th2. The H is for helper. And we have a type 3 helper, Th3. And so the pathogen depends, what pathogen is depends on what type of help you want. So for a type 1, you want a virus or anything intracellular. And this could be a bacteria, it could be a parasite, etc. Type 2, you want a large pathogen. You want an extracellular hemolymph, for example, arthropods. For type 3, it's really important for extracellular bacteria. And this is actually a majority of most bacteria infections. Viruses are always intracellular. Most bacteria are extracellular. Exceptions are going to be, of course, like Mycobacterium tuberculosis, Listeria, and a few others. And so the response, the helped response, is going to be CD8 T cells via cytokines. So remember, CD8 T cells cannot interact directly with CD4 T cells, so CD4 T cells have to communicate, and they release those proteins to communicate with CD8 T cells. Macrophages, and this is direct help, and B cells, and this is direct help, because B cells and macrophages can both interact with your CD4 T cells. The type 2 response is going to be our eosinophils. And this is going to be, again, our B cells and our macrophages. Typically, B cells will make immunoglobulin G, IgG type. Typically, you're going to have IgE for your type 2 responses. And then type 3 is going to be for neutrophils, macrophages, B cells, usually going to be IgG again. Typically when you see IgG, it'll be a combination of IgG, IgA, because IgA will be in the secretions, IgG will be inside of your body. Now, the immune pathology associated with this are going to be autoimmunity. It's going to be the allergic response and autoimmunity. Okay, so that is kind of a big, again, overview of the major concepts, especially with respect to innate and acquired immune responses.